So my little talk today is going to be telling you guys about isolation of bacteria that we've done recently at Kalk Bay. And then I'm going to give a super short crash course in Oxford nanopore sequencing. So hopefully you guys can hang in there. This stuff is way more complicated than I'm going to tell you about. But of course, today really is just about introducing you to the technology, getting your hands on, and the latter part of this, which is the data analysis, we're not even going to do today. But I'll tell you a little bit about that after we finish setting up the sequence runs. All right, so let's get into it. I know you are biology teachers, and I don't mean to insult your intelligence, but I think it's important if you're talking about DNA sequencing just to take one step back and to remind ourselves what DNA actually is. So, of course, we've got these two anti-parallel strands of ribose molecule, nucleic acid. So we've got the DNA basis, and according to Chargraph's rules, thymine always pairs with adenine, cytosine always pairs with guanine. So this is what we would refer to as a DNA base pair. And when we sequence, we always report back how many of these base pairs are we generating. Okay. All right, then I thought it might be interesting just to get a sense of scale. So I just said, when we sequence, we talk about these base pairs. So here we've got a table. It might be very difficult for some of you to see, but it sort of shows the size of genomes from mycoplasma, gram-positive bacteria, gram-negatives, all the way down to the flowering plants, surprisingly, have some of the largest genomes. So here I've just showed, a plasmid is a small circular piece of DNA that we very often find inside bacteria. So there's the sort of size range that you can expect. 5,000 to 150,000 base pairs. Bacteriophages, I don't know if you guys are familiar with those. They are viruses that specifically target bacteria. So again, about 5 kb, 5 kilo base pairs to 200,000 base pairs. Then a bacterial genome, so that'll be roughly 5 million to about 12 million base pairs. And then of course, a human genome is 3.2 billion base pairs. So you can see there's a huge range of sizes that we may want to sequence. Okay, something I think many people might not appreciate is that the University of the Western Cape has actually been at the forefront of next generation sequencing since its inception. So when I joined UWC in 2008, Professor Jasper Rees had just acquired this machine called the Genome Analyzer. And in retrospect, I just give some of the performance numbers for these machines. It actually had a pretty decent output at 25 gigabase per run. So that'll be 25 billion base pairs per run. The reads were exceptionally short. They were only 75 base pairs long. And you can imagine, if you think back to Rob's talk, if I'm trying to reassemble this genome from 75 base pair fragments, it is quite computationally challenging. So fast forward to 2011, and we saw this at a conference at the Lord Charles Hotel in Somerset West, and the eyes went like this. It's like, wow, this is cool. And so Marla and the then director were successful in securing funding to buy one of these for the lab. So you can see much smaller output. The read length now has improved substantially, but this, because we're focused on bacteria, would allow us to finally start sequencing bacterial genomes. This was quite disappointing. We paid 1.4 million for it. That's all taxpayer money. And a year and a bit later, Roche said they're no longer supporting the technology. That was a bad day. All right, let's forget that. And we move on to a very short while later. So Illumina today is the giant. They're the juggernaut when it comes to next generation sequencing. So they bought this company and they turned it into Illumina. So this little unit that came out called the MySeq is the machine that has the largest install base in the world. They sold very many units, and you can see the output. so now we're approaching the sort of output that this one had with a reasonable read length. But the quality of the base calls, the quality of the sequence data that you would get, this is the gold standard today. So of course, this is their baby machine because we're interested in small things, but they obviously produce much larger machines as well. Then we fast forward to 2023, and over the last sort of 10, 15 years, there's a company called Oxford Nanopore Technologies. Their technology is revolutionary. It doesn't do what any of the other guys used to do, and I'll explain to you towards the end of my talk exactly how the sequencing works. And now you can see the physical size of this machine. This is called the P2 Solo, and you can see the output, 500 gigabytes, and the read length, 300 base pairs, 450, 4 million bases long. So this is absolutely game-changing. And, this is almost portable. You can take it wherever you want. Right, so that just gives us some more context. And, that MySeq machine, this is just to brag a little bit. So these are the different publications that have stemmed from our sequencing. So we've certainly put that machine to good use. So that was also bought with taxpayer money. We are very aware that we all contribute to this pool of money, and we try and impart that to the students that these things should count. Okay, so why do we want to sequence a bunch of bacterial genomes? I think there are many reasons, and you heard from Rob. He really explained very well how we can mine it for enzymes and compounds and all sorts of stuff. I'm giving the very basic example. So I think we're all familiar with Alexander Fleming and the discovery of penicillin. So one of the main motivations is trying to find new antimicrobial compounds. I think everybody's aware that bacteria have become more resistant to the different antibiotics that we use over time. And in sequencing the genomes, there's the promise of us finding suites of genes that can potentially give us new antibiotics. It is now appreciated that very many of these suites of genes, and I'm going to talk about it a little bit more just now, are silent. The bacteria don't actually express them when we grow them in the lab. So just like we can't culture the vast majority of bacteria that are out there, we also can't make them express these genes that potentially can be antibiotics. All right. So when you go about trying to find novel compounds, there's sort of two methods today that we can use. The one is the sort of top-down method. So this is your classic biodiscovery, and Rob also alluded to this, where you grow the bacterium in the lab, you let it express its genes and produce the compound, and then using standard chemistry techniques, you go and make an organic extract from that bacterial culture, and you go do mass spectrometry or NMR, and you find the physical compound, and then of course you can decide or test it, is this antimicrobial or not. And if you're lucky enough to have a genome, maybe you can work your way back and say, oh, these were the genes that were responsible for making this. But today, because we have very nice databases and very nice computational tools for hunting for these suites of genes in bacterial genomes, we can take a bottom-up approach. So I can initially sequence the bug first without growing it, and I can potentially identify these suites of genes that can make the compound, and I can go and heterologously express it in some other bacterium if I want. Okay, so just a quick word on a biosynthetic gene cluster. So up here you see the idealized biosynthetic gene cluster. And down here we've just got some structures of various antibiotic compounds that people have described over time. So this is very complex. There are a lot of different classes of these compounds, but essentially all these clusters normally have some core enzymes that'll be responsible for making the main structure of the compound. You might then have some biosynthetic tailoring enzymes that might put a methyl group somewhere. You could have resistance genes. So of course, if the organism is making an antibiotic, it might be killing itself. And so it's got resistance genes, or it's got a transporter to send it outside the cell. because it actually wants to target other bacteria that are competing for resources. You might also have a regulator, so this way the organism can control the gene expression. Right. Cool. So in 2023, we got some money from Diplomix. They've been fantastic partners over the last 3 years. And we went to Kalk Bay. Many of you would be familiar. The idea was to go and sample this guy. So one of the sea sponges that you find growing in the intertidal zone there, as well as this urchin. So this is just a little video showing us sampling. There I am squatting on the rock. And you can see when we scrape this off the rock, we squeeze out the juice. So hopefully you can see in Marla's hand there, it's this brown soup that comes out of the sea sponge. And if you look at that stuff under a microscope, it is teeming with life. So all the way from bacteriophages, bacteria, microeukaryotes, eukaryotes. So there's just a lot of stuff going on in there. The plan here was to do something called single-cell genomics. So I said I'm the manager for the flow cytometry facility. So if you don't know what a flow cytometer is, it is a 10 million rand machine that can single-cell sort anything in your sample. So at single-cell resolution, we can kick out and sort those individual bacteria or mammalian cells or what have you. And you can then isolate their DNA, you can amplify it, and you can sequence everything in there. So there's some amazing technology that we have access to. All right, but the first thing we did, as we always do, is to culture. Because there's every possibility that you've got the right set of conditions for culturing these guys. And Rob also told you earlier that culturing is where it's at. Because if you have the bug in hand, then of course, there's a lot you can do with it. So this slide just shows you the kind of workflow. So, we've got the sponge, we squeeze out that orange juice, plate it on media, incubate, and then we are obviously interested in what they have on their genomes. So we then isolate the DNA, do the sequencing, and then, like I said, we've got fantastic bioinformatic tools with which to hunt for these suites of genes. So this table on the left just shows you some of the bugs. We've expanded on this that we managed to isolate. And yeah, some of these, so if you're familiar with biology, we've obviously got genus and species names for different organisms. So here, for example, the genus name is JAHTBLO1, and we don't know the species. The reason it's called that is we are discovering new bacteria at such a rate, people can't keep up with naming them. So, the whole next generation sequencing business has been an absolute, mind-blowing experience for microbiologists in particular. And down here, it doesn't show up very nicely, but just amazing morphologies that you see on a plate. Okay, so if we then come back again to these biosynthetic gene clusters, here, and I appreciate this doesn't immediately mean anything to you, but so these black arrows just indicate the individual genes in the cluster. The little round circles indicate different domains, protein domains that are responsible for certain functions. And what you may appreciate from this is that it looks like a repeated pattern. So these three are examples of non-ribosomal peptide synthases, and the class of compounds that they make very often can be antimicrobial. So these three that I highlight here are brand new. They've never been seen in a database. They come from Kalk Bay, a sponge intertidal zone. And this is just one exercise. If you take that South Africa has a massive coastline at, 2,000 kilometers plus, there's a lot out there to go and discover. So the main database that we compare these two is called MyBig. It's the same folks who wrote Anti-Smash. They also curate this database. And over here, I just show sort of a breakdown again. I appreciate this won't mean much, but the various classes or families of these type of compounds, there's a lot of interesting stuff in there. And we can also do some sort of network analysis. to compare the various pathways to one another. So this seems quite simple. If there are lines between the dots, that means that these pathways show some kind of relationship to one another. And if it's a dot on its own, that's a singleton. It means there's nothing like it in a database, and there's nothing else similar in the same data set. So a lot of unique stuff. So the other thing we can do with, once we've got the genomes, is we can compare these guys to one another. So what I show here is an example where this is, so Rob showed these massive phylogenetic trees. So this is, if I now zoom in on the tree, I can see here I'm comparing 3 bacteria, S94, S1, PE1407. And this tree was generated by a program called Genome Taxonomy Database. What this does is it takes 120 genes off the bacterial genome, it concatenates them, and then compares them. And at the moment, they've got something like 105,000 different bacteria in their tree of life, bacterial tree of life. So when I do this exercise with these 3 bacteria, essentially they come out as the exact same species. So if there's a branch length, then it signifies that they are different. These guys essentially lie on a straight line, indicating that if given those 120 marker genes, they are identical. But if I compare the whole genomes, I can see that there are large portions of these genomes that don't match. So this is the so-called pan-genome. So even though you think you're dealing with the same bacterium, you are really not. And very often it's these segments that are different that encode these weird and wonderful biosynthetic gene clusters that we're after. Okay, so now I'm going to switch over to talk about the actual sequencing. And again, just to remind us, what we're trying to do is we're trying to determine the sequence ACTG when we're sequencing the genome. So I've just got a video here. This might go on this side. So this hopefully should give you some idea of how this technology works. And I'm just going to talk talk us through the video. So this little device is called the Mark 1B. It's the smallest machine they make. And that sort of black chip, you're going to see it in the lab, is called a flow cell. So there's a very small window right there where you've got nanopores, and you'll see them now. They are physical proteins that sit in a membrane. So what's shown here, these little fuzzy squiggles, is the DNA, your DNA library that you've created. There sits the actual pore. And onto your DNA, during the library creation, you attach a motor protein. So what the motor protein does is it unwinds the double strand and it feeds the single strand through the pore. And you'll see now that as the DNA moves through the pore, there are electrolytes that move through, and that obviously creates an electrical signal. This pore protein will dance around, given the sequence that's busy being pulled through the pore, and an electrical signal is generated off that. So that electrical signal can then be interpreted with a machine learning algorithm to convert the electrical signal to your base calls, AGCT, what. That it is mind-blowing technology. Like you're going to see the thing and you're like, what? This is it, you know? But the road to developing this technology, it's amazing. I need to become an Oxford Nanopore salesman because this is like the coolest stuff out. So yeah, just again to show you the 2 main features of this is the DNA going through the pore, an electrical signal gets generated, and that gets...
Stupid question, but how many samples are you putting in there in one go?
You can multiplex. Yeah. You can put up to 96 samples together. And each one gets a unique barcode. So after the fact, we can pull them apart. Right. So just to show you, you're going to see this machine in the lab. So they've made several different form factors. So this is the big daddy, the Promethean. Then they've got the Gridiron, the P2 Solar. So this guy is what I showed you earlier that does the 500 gigabase. So it takes 2 flow cells. This one takes 48. So if you're sequencing human genomes, then you're using this. So down here, I just show examples of the 3 different flow cells that they sell. So this is the Minion flow cell, and that'll go into the machines that we're going to use today, called the Mark 1C. And then those are the Promethean flow cells. Okay, so the workflow in general is this. We've got a sample. So Stephanie and Chelsea have done a lot of hard graft. They've isolated some new bacteria also from the same sponge, and those are the ones that we're going to be sequencing today. So we have no idea what these bugs are. You guys are going to be generating the data so we can figure this out. You've got your sample, you want to isolate some DNA, and today there are fantastic kits for doing this. Yeah, the whole space has come a long way because 15, 20 years ago, metagenomics wasn't really a thing, and isolating DNA from weird and wonderful bacteria also wasn't really a thing. And microbiologists in general don't get a lot of love from these companies. But now the kits exist to do that. Of course, you want to do the sequencing. That's what we'll do next. And then after we're done, we'll come back and I'll talk about some of the analysis. How do you deal with the data? What can you do with it? So again, this is the crash course. What are we going to do? So the kit that we get from Nanopore for creating the sequencing library, it contains an enzyme. So that is, the diagram trying to depict the enzyme over here. And these lines, the red and yellow lines, they are pieces of DNA attached to the enzyme. This enzyme can hop into your DNA and break it. And when it does that, it attaches those pieces of DNA to the genome. So this is attaching both the barcodes and adapter sequences all in one go. We'll then add the adapter sequence with the motor protein loaded on it. So if you cast your mind back to the video, that purple protein, the motor protein, and your fragments of DNA, so it's essentially going to chop up our DNA, will then look like this. So the green in the middle, that's the genome you're trying to sequence, and then you've got the barcodes at either end. And this is the way that we can, after the fact, identify which sequence comes from which organism or which sample. All right, I'm getting the signal for time. Okay, but I just wanted to give you an impression of what is going to happen, because I don't know who has a background in molecular biology or if you've ever been exposed to anything molecular biology-wise. I know you do. No, you do as well. But what you're going to see is a plastic tube with 10 microliters at the bottom. But I want to give you, again, a sense of scale. So the question is, how many DNA molecules, how many copies of that bacterial genome do you actually have in that 10 microliters? So here we've done the calculation. And it turns out there's about 36.5 million copies of arguably a 5 million base pair genome in that 10 microliters. So keep that in mind. So when you see that, it's not just one copy of the bacterial genome. That's the number of copies. Okay. Let's go sequencing. We've now done this sequencing, and the question was already asked, what do we do with the files once we get it? So, yeah, here's my very simple workflow, and we're at the last stage here. What do we do with the analysis? So naturally, some questions pop up, like, what information do we want to know about our sequencing run? One of the questions could be, how much data did we get? So we spoke about that earlier, and you can measure that in megabases. Hopefully that's not what we get. We get some gigabases. What was the N50 value? And I'll explain what the N50 value is a little bit later. What was the average read length? So the longer the read, it makes it a lot easier computationally to put the thing back together. So a bacterial genome is essentially a circular piece of DNA. So just imagine a string that's, circular string. So for very complicated genomes, it's like really beneficial to have these super long reads. And nowadays with Oxford Nanopore, they actually, if you guys know what a telomere is, so that's these sequences at the end of a human chromosome, they're talking about telomere to telomere assemblies of human genomes. They can finally do that with long read technology. What are the numbers of reads that we got? What was the quality of those base calls? So one of the drawbacks of Oxford Nanopore, maybe still until 2-3 years ago, was that the quality was not as good as, say, Illumina. But that has changed, and today it's on par with Illumina. Did my barcodes sequence evenly? So we put in 20 bacterial genomes, but maybe there's one superstar that sequenced really well and another one that's a bit of a dud. So once the run is finished, one of the output files that you get is an HTML file that contains a lot of that data, or those will answer those questions for you. So here I just show you sort of screen captures of that output. So, in terms of raw data produced, this particular run produced 124 gigs of data. It tells you how many of them failed the quality filter and how many passed. You could also look at the read length distribution. So in this case, the N50 should be here somewhere. No, I don't see it right now, but most of the reads sort of cluster around 3 to 10 KB kilobases. You can also see how the run progressed. So the green line are the reads that pass our quality filter, and the red lines are the ones that fail. The blue line is the total number of reads. And so essentially you can see that over the course of 45 hours, it starts off, you get most of your sequence data within the first 40 odd hours, and then it starts to level off. So this shows you the performance of the pause over time. So you can see at the beginning, we've got a lot of pause, everybody's happy in sequencing, and then as the run progresses, they get tired and they sort of peter out. I'm racing through this because, I'm sure we all want to get home at some point. So where do we do the data processing? If your data set is pretty small, you can do it on a local, pretty powerful laptop even, or a powerful desktop computer. So one thing that happened quite early on with Oxford Nanopore is that the people in the community, they farmed these machines out to academics all across the world to test the technology. And some of those said, but the processing, converting that electrical squiggled signal to base calls was super slow. And the academics actually wrote software to take advantage of gaming graphics cards to do the base calling. And it's orders of magnitude faster. So today, all the base calling happens on GPUs or graphics cards. So if your data set is really big, then I don't know if you guys were aware of this, but South Africa makes available to academics some computing infrastructure through the NICI's, the National Integrated Cyber Infrastructure System. The Center for High Performance Computing is hosted in Cape Town. And so we can access that. We can upload our data. You can ask them to install software, and all of the data processing can happen there. They also provide a place to store all of this data. So if you're sequencing human genomes and so on, the data sets grows very large. So the RESA is the storage facility that they make available. So for IMBM, we've organized 100 terabyte of storage there, and you can upload as much as you want, and they've got proper cold long-term storage as well as hot storage, so you can work with it in real time. Okay, but in practical terms, we've now got this electrical signal from our sequence run. What do we do with it? So this diagram just kind of shows you the process that the algorithm goes through to convert that electrical signal into base calls. Oxford Nanopore have named all their software for fish names. I don't know why. So the initial base caller was called Guppy. The next iteration is Dorado. And so, yeah, this is just conversion of the squiggle, that electrical signal, to base pairs. So that's step one. The next thing we do is to demultiplex. So we have now pooled, 10 genomes with barcodes, and we've mixed them together. How do we pull them back apart? So that's the process of demultiplexing. And again, the Dorado software is what we use to kind of go through the motions and then end up with our individual genomes. Okay. Yeah, so this is, again, just to remind us of how we created the library. So the next step would be to trim off those barcode and adapter sequences. So this is all done in software, and we use so this is not an Oxford Nanopore software, but this one is called Pore Chop. You can see people have a lack of sense of humor. Anyway, so at this stage now, we're removing these extraneous sequences just to retain the actual sequence that we're interested in. Another pre-processing step that we can do is just to remove any low-quality reads or particularly short reads. So those aren't helpful to us. So again, we can tell them anything less than 1,000 base pairs we want to throw away, and anything with a quality score less than 10, chuck that out. So this is all like getting our base calls, pre-processing the data before we do anything with it. And if we then want to, we can go ahead and try and recreate our genome. There are very many of these assembly programs, but Fly in recent years has come out as #1 for long read assemblies. I'm not going to pretend to know exactly how it works. This is all, bioinformatics. I'm a microbiologist by training. I am a weekend bioinformatician, call it that. But yeah, so this is the program we use to reassemble the genomes. And until recently, the last polishing step was error correction with a program called Medaka. This is no longer needed because the quality of the base calls has become so good, and the quality of the software for doing the base calling. All right, so other things that you may want to do is just to plot your data and get sort of a visual of what's going on. So here we're plotting read length versus the read quality. And you can see in this case, this data set had a very many high quality reads. All right, so if you now pull all this information together, then you can start putting tables like this together. So here we've got some of those Kalk Bay isolates that we sequenced. So this was the first batch, and then you've got your read length N50, N90, the number of reads, the GC content, read length, yeah, a lot of different things that you can do. All right, so now that I've reassembled my bacterial genome, some other questions pop up. So which bacterium did I sequence? What is the genus and species? How is this related to other bacteria? Or did I find something completely novel? Which genes or proteins does the genome encode? Was the DNA I extracted contaminated with that of another organism? And how bad is it? So sometimes if we're growing the organism in the lab, I don't necessarily have a way to control, except for my sterile technique, that some other bug didn't fall in there, and now I've got a mix of two bacteria. So we need to have a way to figure this out. Which secondary metabolite gene clusters does it have? So earlier we talked about biosynthetic gene clusters, and I may want to check those out. What extra chromosomal elements does it have? Does it contain plasmids or chromids or anything like that? Right. So to answer that first question, did I sequence a novel bug or how does it relate to anything else? The tool that we use is Genome Taxonomy Database. So these folks have done a fantastic job, and they continue to incorporate more and more bacteria as we find them. So as I said earlier, 107,000 bacterial species in their tree of life. takes 120 marker genes, concatenates them, compares them all, and then sticks them somewhere in that tree. So you know in fine detail if you have a new bacterium or not. This is something I mentioned. Oh no, I didn't mention this. So if we want to annotate the genome and see, so each one of these little bars over here is a gene, we use a program called Bacter. So we just chase it through the program. It calls the genes where they are. It compares it to a database and annotates the genome beautifully for us. So the tools have really developed very nicely along with next generation sequencing to enable us to quickly come to grips with these genomes. So in terms of contamination, we've got a program called CheckM. So we'll run the genomes through there, and this is sort of the normal output that you get. But the two things that we're really interested in is the completeness and the level of contamination. So again, you get a very good sense of whether you've got a single genome there or there's something else going on. The long read technology actually makes it so trivial, even if some other bug fell in there and you sequence the two together, after you've assembled, sometimes I've ended up with two complete genomes, and I could literally just copy the one and paste it into a different file, and I've separated them. It's that trivial. In terms of secondary metabolite gene clusters, this is the go-to program for all people looking at secondary metabolite clusters. Anti-Smash, they keep updating it, they keep broadening the database of known characterized pathways. So I appreciate that because we're not physically doing this, much of this stuff maybe doesn't register or doesn't mean very much, but yeah, so the tools are all there for us to figure these things out. Okay, so how does sequencing enrich a biobank? So I've just listed 4 things here that I think is most important. Just to mention the 2 biggest culture collections in the world, that's the American type culture collection and DSMZ, both have now started sequencing all the bacteria in their collection, and those genomes are also available to anybody else. So from my perspective, the way that sequencing helps the biobank is, one, to de-replicate strains. So you don't necessarily want to carry 1 million copies of the same bacterium in your biobank. It immediately becomes a resource for microbial ecologists. So Rob's talk earlier gave you a fantastic intro into how you can use microbes to kind of say anything about an environment. It also becomes a resource for biodiscovery. And again, Rob alluded to that. So if you want to find new enzymes, want to find new compounds, it's fantastic for that. And then for me, lastly, and this is really for us, training. This is just a portion of the folks who we've trained on ONT sequencing over the last 2-3 years. And again, it's all with the help of Diplomics. So, we use it as a vehicle to get people familiar with NGS. And at the same time, they're sequencing these genomes and it becomes a resource for everybody. So it's been a fantastic effort. Okay. So going beyond the biobank, or not really, so Rob explained this earlier, this is just a different way of doing that, and that is these metagenome assembled genomes, where we go and grab an environmental sample, we extract all the DNA, we sequence everything, and now in software, computationally, we try to piece together the microbial genomes. Now there's no barcode to help us with that. So again, bioinformaticians are quite clever and they've given us the tools to do so. So again, we've got a mix of bugs, we extract all the DNA, we make a library just as you have, we sequence, we assemble those pieces, and if you remember Rob's puzzle analogy, we always lose pieces. So you never get the whole thing back, but that's okay. So then we do binning. So this is the magic of trying to separate out the genomes. And then we can go on to run through those tools that I just described. Do I have a novel bug? What pathways does it encode? So on and so forth. So yeah, there is Marla and me and one of our daughters. That was 2 weeks ago in Kalk Bay. So thankfully, the Plowies gave us some more money to try and get DNA out of this sea sponge that is defied DNA extraction for a year and a half. So yeah, there you can see the brown juice as we always get it. This is what it looks like after I try and filter out all the eukaryotes and things we don't want to leave us just with the bacterial fraction. So yay! After very long, we have DNA on a gel. And I did the sequence run last weekend on our P2 Solar back at IMBM. And yeah, we got 40 gigs of data that passed filter, and it looks like we've got some really interesting bugs in there, predominantly anaerobic bacteria. So those are the guys who grow in the absence of oxygen, which we would never be able to culture because we don't have the facility to culture them back at the lab. Yeah, so early insight into that. So we will now start to understand, at least for this sponge for now, and then later we'll broaden it out to other species, samples. What are the bugs that are in these things and what do they do? Blank slide. I just want to give a big shout out to Shane and Tim for continuing to partner with us. And I encourage you to partner with them in any way, shape or form that you can. It's been a really fruitful relationship. So thank you very much.